All right, I'm Mark Ziolo. I'm in the same department as Dr. Jansen, physiology. We're also in the Heart Lung Research Institute, and both of our labs are in Hamilton Hall. So my lab is 1,000 square feet, except I am moving to Graves Hall next month, but I was told I was moving 6 months ago too. So I don't know how exactly big my lab's going to be or anything like that, but eventually we'll be in Graves Hall. Right now I got 6 members in my lab. I have a postdoctoral fellow. I got 2 students in that undergrad College of Medicine program. I got 3 undergrads, and unlike Dr. Jansen, I think mice can give us a lot of information. So we have a huge mouse colony that the undergrads help take care of, and I do have a slot for 1 graduate student. I am funded as well, and money shouldn't be an issue for the next 3 or 4 years. Rotation. For those of you interested in rotating the lab, I need blocks of time. So I know with your classwork, you don't have to come to the lab every day, but I do need a good chunk of time. Like Dr. Jansen, I'm a physiologist, so these experiments take lots of time, hours, 4, 5, 6 hours at a time. So what I like to do is, my philosophy is see one, do one. You get thrown right into the lab. Don't worry, you can't break anything in my lab. The stuff that you can't break, we'll send you to Dr. Jansen's lab. All right? So don't worry, nothing can break. You'll get thrown right in and start doing experiments. With the small lab, I do spend time with you. I'm here all day. I don't have anything better to do, so I'm in my office. My door is always open. You guys can come in, talk, science, whatever. What do I expect? Graduate within 5 years, and that's from joining the program, not joining my lab. Just like Dr. Jansen, you'll have your own project. You will submit a grant on that project. Classes, we are physiologists, so our department's got a physiology class that you'll take. We have an advanced cardiac physiology class that you'll take. We have cardiovascular journal clubs, so your classwork is kind of mapped out. Same thing as Dr. Jansen. I'll send you to 1 or 2 meetings, the American Heart Association, International Society for Heart Research, Gorn conferences, biophysics. There's a whole range of meetings. Hours. I don't care about hours. I want papers. All right? 1 to 2 papers a year. If you can do a paper a year working 2 hours a week, God bless you, good for you. If it takes 60, 70 hours a week to do it, 60, 70. All right? I don't keep track of hours. I want pubs. I've had 4 graduate students. They've had a total of 34 pubs. Same like Dr. Jansen. They've gotten HA and NIH predoctoral fellowships. They've won awards, department awards, HLRI awards. They've gotten national awards from Gorn conferences, international awards from American Heart Association. 3 went on to do postdocs in good labs. 1 went to industry. And actually, my first graduate student's now an assistant professor at Johns Hopkins. So I think I've done a pretty good job with the graduate students. Okay. OSU, we're cancer. I have to tell cancer people this all the time. Heart disease is the #1 cause of death in America and the world. You add up the death from the 7 who's here for cancer? See, add up the death from the top 7 cancers doesn't equal heart disease. So what does my lab want to do? We want to take the sick heart, give some kind of therapeutic intervention, and make it strong. So that's what we're looking at. We know how the heart works. We know the major players. We know a lot of what's gone wrong with the bad heart, the diseased heart. So, oops. So here's a picture within a muscle cell, a myocyte. Or not. Why is this not working? Oh, never mind. But as I said, we know the proteins. Although there are some differences between mouse and human, many of them are the same. So we can look at the individual protein function through various techniques. We use patch clamp, we use microscopy. So individual protein, we can look at the isolated myocyte. Here's the muscle cell. This one we can measure electrical activity. Calcium is what makes it contract. We can measure how much it contracts. We can measure in vivo cardiac performance and function. So we can do echocardiography on a mouse. We can stick a catheter inside the left ventricle of a mouse and get pressure-volume relationships. We can put them on a treadmill, do their VO2 max, how much they run. It's always fun running mice too. All right, so we want to look for therapeutic inventions. The diseases we're looking at, myocardial infarction, MI, we permanently ligate a vessel, give them a heart attack, and try to throw on some therapeutics. We have a pressure overload model, obesity. This heart disease, it's only going to get worse. As the population gets fatter, that's heart disease. As the population gets older, that's heart disease. So we have obesity models, we have diabetes models, we have aging models. And as Dr. Jansen did point out, not everything that works in the mouse is going to work in the human. So in collaboration with Dr. Jansen, we do use his human hearts to see if we can translate some of our findings. Here's an example. This is a interventricular pressure through our catheter. Here's systolic function, here's diastolic function. As we increase heart rate, systolic function increases. Here's a mouse with the myocardial infarction, the heart attack. You can see systolic functions decreased. Here's a drug we termed Demepo that was developed here at OSU in collaboration with some people at HLRI. Just acute administration of this drug, we can increase systolic function, and same thing. Diastolic functions here is bad in the MI, and then with this drug, we can increase diastolic function. Drug discovery, some big projects that we're working on right now is brown adipose tissue. They release signaling lipids termed bradykines. We're looking at the effect of these bradykines on heart function. So here we're given, once again, catheter. We cath the mouse, measure systolic and diastolic function. Here's with this bradykine. We can improve systolic and diastolic function, and we're trying to discover some of these bradykines if they are going to be protective in the diseased heart. We also do gene therapy. This is in collaboration with Dr. Davis, another member of our department in physiology. We give a mouse an MI. 2 weeks later, we inject AAV with the mutant protein. This is using echocardiography. Here's EF. And as we get expression of the protein, you can see that we can increase heart function. So we have a lot of different interventions that we can look at. Hopefully some of them will turn out to be therapeutic. Here's my contact information, and I'll take any questions as well.